Assalamu alaikum and good afternoon, everyone, and welcome to today's webinar on biosimilars challenge in developing and regulatory approvals. My name is Dr. Naveen Sanyl, Manager Regulatory and BD, and I will be your host for this session. Today's webinar is focused mainly on We are honored to have attendees from across the healthcare and pharmaceutical sectors joining us. We are privileged to have Dr. Adeel Shahzad, Regulatory Specialist, Biologics, National Health Regulatory Authority, Kingdom of Bahrain with us today. Dr. Adeel Shahzad holds a Doctor of Pharmacy degree and has completed numerous specialized courses and certification in biosimilar, advanced therapies, bioethics, drug discoveries, and development. And regulatory affairs from STEAM institutions like the University of Oslo, Harvard Davidson, and UOB. With over 13 years of experience in the pharmaceutical and biopharmaceutical industries, Dr. Adeel's expertise encompasses regulatory affairs, manufacturing, quality operations, product development, GMP compliance, and pharmacovigilance. Since joining NHRA Bahrain in 2022 as a biologics specialist, Dr. Adeel has taken on responsibilities including the assessment of new registrations, conducting GMP audits, and training on biologics assessment and guideline implementations. Actively in participating both the regional and international joint regulatory working groups. Dr. Adeel contributes to the preparation and revision of regulatory guidelines. Before we will be having 10 to 15 minutes dedicated for Q&A sessions at the end. Feel free to post your questions in the chat anytime. Let's get started. Dr. Adeel, over to you.
Thank you for having me, Naveen. Assalamu alaikum everyone, and आप सबको रमज़ान बहुत-बहुत मुबारक हो. First, I must appreciate the effort by IBBPS for establishing this learning platform, especially for pharmaceutical learners and the professionals. I understand it's a Ramadan and we are not in the fullest of our energy, so I'll try to keep this session as crisp as possible. So in this session, we'll start with the basic understanding of biosimilars. Then we'll talk about the global regulatory requirements of biosimilars. And we'll also discuss what are the challenges a company faces while developing and seeking regulatory approvals after the development of their biosimilars. So let's just start because I have a very short period of time. Naveen just gave me 30 minutes to finish this presentation, but I'll try to catch it up within the specified time. So some mandatory disclosure that I have no commercial affiliation or financial interest. The presentation is for informational purposes only. If you see any brand names or the pictures of any branded medicines, this is only just for the educational purposes. And the views expressed are my own and do not necessarily reflect those of my organizations. Well, before we go into nuts and bolts of these biosimilars, I have a very interesting fact to share with you. Actually, it is something that we can ponder upon. As you see that the statement says that all biosimilars are biologics, but not all biologics have or can have biosimilars. Well, देखने में तो ये बड़ी simple लगती है. The first part is quite simple, and actually it gives us the basic definition of biosimilar, very simple definition. That what are the biosimilars? Actually, they are just the copies of innovator biologics, जिनका patent expire हो चुका होता है. Just like the generic medicines, जो copies होती हैं innovator synthetic drugs की. But if you look at the second part of this statement, that is a bit tricky, and it gives you a lot of insight about the biosimilars. It says that, but not all biologics have or can have biosimilars. Well, to understand this, we have to understand that what type of categories of biological product have biosimilars in the market. So here are some of the examples. Most of the biosimilar you will find in the market are monoclonal antibodies or the MABs. For example, Humira, it's an adalimumab, which have many biosimilars approved so far, like Emcevita, Idacio, Helio, Hyrimoz, etc. And the other category which have biosimilars are the insulins. Like we also have been biosimilar for Semglee, for insulin glargine. Also, we have approved biosimilar for Lispro. Another category are the hormones like filgrastim and the ILIA fusion protein. They also have approved biosimilars. Epoetin alpha, recombinant erythropoietin, has a biosimilar direct, and enoxaprins have many biosimilar approved in Europe. So just look at these approved biosimilar, you'll find that either they are monoclonal antibodies, recombinant proteins, or the fusion proteins, or the hormones. But this is not the only categories of product that we call them the biologics. There is a huge category. For example, the blood and blood-derived products, vaccines, allergenics, gene and stem therapy products, tissue engineering, enzymes, toxins, and many others. But the question is, have you ever seen any biosimilar of any blood product like whole blood or the RBCs, platelets, or any blood derivative products like the albumins or the factors, clotting factors, or even for the vaccines? No, not. The answer is no, not yet. But why is that? To answer this question, you will find in the later part of the presentation. So I'll ask you to keep focusing on the key points in the presentation. And when you find the answer, I encourage you to write it down in the chat box. We'll discuss it at the end of this presentation during the Q&A session. Well, when we are talking about the biosimilars, actually we are talking about the biological products. So we have to understand some basic characteristics of biological products. So what makes a biological is actually they are derived from the natural sources. Either we extract them or we synthesize within the living cells. They can be extracted from humans, animals, mice, like mice or the microorganisms, or can be synthesized within the cell lines of mammalian, avian, insects, and the plant origin. Another characteristics is for the medical use, they are used for the prevention, cure, mitigation, diagnosis, and treatment of many diseases. And nowadays, it is on the biologicals are on the forefront of treating many diseases. Like in oncology, you'll find it in immunology, endocrinology, and in vaccines, and many other areas where they are flourishing well. One of the most important characteristic of a biological product is that they are generally very large. Large means in comparison to small molecule drugs like the synthetic drugs. For example, aspirin, as you see in the table, they are very big. You see the difference between the size of an aspirin, which contains only 21 atoms, and the difference in the size of a large monoclonal antibodies, which may have up to 25,000 atoms. Yes, so you can see that the difference is huge, and maybe the biological product can be 1,000 times bigger than our synthetic counterparts. So the size of these biological molecules actually makes them delicate and fragile, and we see that they are more prone to like heat and the physical stress. They are not only large in size, but also they are very complex. Just a comparison of how complex they can be, you can see that I have compared it with a very rudimentary bicycle with a very high-tech jet plane. So you can understand that how complex these biological products can be. Yes, biological can be expensive, and when I say expensive, they can be insanely expensive. Just to give you the example that how expensive they can be as expensive as buying a Ford Mustang. I'm not kidding. And here are some of the examples of prices, international prices of some of biological products. As you say that the Exotase Alpha is like you buy it in $73,000, and other biological product you see they are like 20,000, 30,000, 22,000 US dollar. So they can be very expensive. And here I'm not talking about the gene therapy products. Well, they are insanely high prices. For example, the Hemgenix, LVDS, and Casivy, they can be as expensive as 3.2 million or 3.9 million dollars. Yeah, so this is why these biological products uh, are unique in nature. Like its access to the general public is a challenge for not only regulator but also for the governments who are investing in for the therapies of these biological products. So, because of this expensive nature of these biological products, the terminology biosimilars came in. They also known as the follow-on biologics, similar biotherapeutic products, subsequent anti-biologics, similar biologics, but generally nowadays all the regulators call them biosimilars. The need for a biosimilar started after at the end of 1990s when the patents of many biologicals started to expire. Then there was a need that we need a copies of these biological products. So there was no regulatory framework for these the approval. So EMA has started working on it, and ultimately in 2001 they published their first draft guidelines, and they called these products the biosimilars. And later on in 2006, first biosimilar product was approved by EMA, Omnitrol. Then Japan and Canada adopted the EMA guidelines, and the US was very late in the whole picture of biosimilars. The first guidelines was actually published in 2012, just after the BPCI Act was passed by the government. And the first biosimilar approved by FDA, US FDA, was Zargio in 2015. Since then, there are many biosimilar has already been approved. EMA has till date approved 126 biosimilars, and US FDA has already approved 71 biosimilars. As I told you, there are different categories of biosimilars which have been approved. Mostly, which of these are monoclonal antibodies, fusion proteins, hormones, and such recombinant proteins. And look at the number of these approvals till the inception of the concept of biosimilars. So now we are expecting more surge in the approval of new biosimilars, because in this decade, a lot of biological products patents are going off. So the global standards, the three regulatory authorities have their global standard which are being followed across the globe. Like the European Medicine Agency, EMA, they are pioneer. They have they have overarching guidelines on biosimilar, also some product-specific guidelines, handy guidelines, and flexible as compared to the US FDA guidelines. US FDA has more customized guidelines as per their US framework. And yes, we also follow these guidelines. We get a lot of beneficial insights about the biosimilar. WHO has a global guideline which gives us the broader concept of the biosimilars, which are being followed by most of the countries. So all these guidelines have similarity between them. The basic concept, the requirement of the documentation are same. The only few minor differences are there, which I'll discuss later part of this presentation. Each authority has their own definition of biosimilar, but they are same. Just to dissect this definition, I have some key facts that what actually a regulatory authority demands from a biosimilar. But the definition says that a biosimilar is a biological product. We know that. And a biosimilar is highly similar to a reference product. This is very important. This is the basic demand of a regulatory, that a biosimilar should be highly similar to a reference product. Another demand or expectation of a regulatory that a biosimilar should have no clinical meaningful difference from a reference product. And a biosimilar is approved by the rigorous evaluation and by the product developers. This question always arises in our mind when we talk about the biosimilars. That why biosimilars are called the biosimilar and not the generics? Well, the answer to this question lies in the structure of these protein molecules. Just look at the structure of these large biological molecule. It's very untidy and it's messed up. But when you compare it with the small molecule drug, it looks very like simple and well-behaved structure. Therefore, the replication of a small molecule drug or a synthetic drug is possible. So we can make generics out of it, and we are already making it through the chemical synthesis. But do you think that exactly copying this large molecule of a protein exactly in the same way? No, it's not possible. Because these biological products are actually produced in living cells. And the living cells have their inherent ability for variability of the proteins. For instance, when each cell can make exact copy of proteins, but you see that after the translation in the ribosome, the protein undergoes post-translational modification. The further and further modifications, like the glycosylation, hydroxylation, bondings, foldings, methylation, and all these post-translational modification are actually the culprit of what we call why we can't make exact copy of biosimilar of the biological product. So because it is uncontrollable, and due to this uncontrolled nature of biological systems and these products, as a regulator, the regulatory authority accepts these variations and heterogeneity. Therefore, a unique set of guidelines were prepared for the biosimilars. So this inherent variation actually tends a biosimilar to have multiple different copies. Not only they are different from their counterpart reference product, but also when you manufacture different batches at a different facility, they can also have a different set of proteins in it. And also even a single vial or an ampule of a biological product has maybe hundreds and thousands of copies which are different from each other. So we look at, just understand that these variability is so immense that we cannot control it. And furthermore, the second thing that that makes it hard to make an exact copy of these protein molecules is our manufacturing process. Uh, This manufacturing process, actually, this is we can say that the layout of a manufacturing process of any of the monoclonal antibodies or fusion proteins or the biosimilars. Now they have become a platform technology, and we have much information about it. So the manufacturing process is also very unique. You see that it is divided into two parts, like the upstream process and the downstream process. And it involves a lot of sophisticated technologies, equipments, and you need a lot of controls to manufacture these products. In upstream process, that starts with the working cell bank. And what is working cell bank? Actually, working cell bank is derived from the master cell bank, which is actually the seeding of the whole biological product, and it remains same throughout the life cycle of the product. So these master cell bank actually contains the recombinant expression system or the recombinant host cell lines, which has already been developed during the research and discovery phase. And now they have those seed cell lines which they use it throughout the life cycle of the product. So through master cell bank, we have the working cell banks, which are thawed and then expanded in a culture media in a seed terrains, like the many seed bioreactors, until it reaches to your required production capacity. Once it reaches to the production bioreactor, the protein is then separated from these cells through the centrifugation, we call it a clarification, and then it has to go through multiple chromatographic columns for the purification. So there are very sophisticated chromatographies are used, like the affinity chromatography, ion exchange, cation exchange, hydrophobic interaction chromatographies, or multimodal chromatographies. are being used. Then once your product is purified, it undergoes the filtration process. Then it's filled into the primary container and then stored at the recommended temperature. There are so many in process controls and process parameters involved in the production of these biological products. So any variation in a very slight variation in pH, temperature, dissolved oxygen, or the culture media, the nutrients which you are using may change the nature of the protein and can change the inheritance or the heterogeneity of the protein. So the controlling the manufacturing process is also very challenging, and we cannot control at an optimum level. So you have to in place a lot of control strategy to make that to ensure that every time you manufacture a protein, the batch has more similarity or the similarity with the one which has already been approved by the regulatory authorities. The biosimilar and the biological regulatory pathway is different. Like when a biological or an innovator drug is innovator biologic is submitted for approval in regulatory authority, it has to go through all the product development stages, starting from the discovery, development, extensive preclinical studies, extensive phase 1, extensive phase 2, and the phase 3 studies on all the indication they are intending to approve. But and of course it costs like more than $1 billion, and a lot of time has to spend to develop a biological product. That's why they are very expensive. And when it comes to the biosimilar product, when biosimilar is submitted for the approval in a regulatory authority, it doesn't have to go through all the development stages. The pathway for biosimilar is abbreviated. It's shortened. We do not need to go for the discovery, and even during the development part, they just have to compare their product with the reference product, what we call the analytical similarity assessment. Preclinical part is very reduced. Only we require it when there is some differences shown during the similarity assessment. Phase 1 trials has to conduct where they compare their product with the reference biological product, and phase 3 studies which they only have to show the efficacy and safety is comparable to the reference product. Only any one of the sensitive indication. They don't have to conduct phase 3 trials on all the indication which reference product has been approved. So they can extrapolate later based on the certain criteria, which I'll tell you at the end of the presentation. So that not only the cost of the product is reduced, and also the timelines of the approval and development of the product is reduced. So the regulatory approach for biosimilars, for originator biologics, we have a higher regulatory emphasis on the clinical studies, later clinical studies like phase 2 and phase 3 clinical studies. We just look for it whether the product is actually efficacious and safe in the patient population. It has any positive risk benefit, and then it's been approved. But in case of biosimilar products, this whole parameter is ups and down. Like our major emphasis is not on the clinical study. The major emphasis on how you are comparing your product in your lab, like on the basis of the quality, on functional characteristics, physicochemical properties, how the structure is being compared. So our major concerns and the focus is on the analytical similarity assessment rather than you have to show the efficacy and safety in the clinical studies. So there is a term used like totality of evidence in US FDA and EMA call it the stepwise approach. There are 4 major things on which a biosimilar product has to show that they are comparable to reference product. The first one I mentioned earlier, that is the analytical similarity assessment. The analytical similarity assessment, you have to show in the lab that your product is comparable by conducting a number of quality tests. Then if analytical similarity assessment is successful and you have seen some of the minor differences during this assessment, you cover those differences through the non-clinical comparability. If these are successful, you submit your application as an IND in the regulatory authority, and you ask for them for start the clinical trials. Then you go for the phase 1 clinical trial where you compare your product with on the basis of PK and PD. And if they are successful, then you move towards the phase 3 and the last stage of the product development. The only expectation what we have as per the definition of biosimilar that your product should be highly similar and there should be no clinical meaningful differences than the reference product. The first step, the analytical similarity assessment. Well, this is, I am again emphasizing on this part because this is the part where the company has and the regulatory authority has a tug of war, because there are a lot of things that regulations demands and the company has to fulfill. For example, they have to identify hundreds of quality attributes of their products and then sort it out on the basis of criticality risk assessment that which quality attributes are actually critical in nature. And then they have to characterize those quality attributes, the critical quality attributes, extensively. For extensively means they have to purchase a lot of reference product from the market, and they have to manufacture number of batches of their biosimilar product, and then they have to compare it head to head. And the techniques and technology what regulatory demands it should be orthogonal. Like if one quality attributes you're testing, you have to test it with the different equipments and different technologies to ensure that you are doing it right. Why we are doing asking for this approach? Because most of the analytical techniques, especially the bioassays, are highly sensitive and they are not well developed, and they are developing with the passage of time. And it's, most of the time, the companies or the developers develop their own analytical techniques or the analytical method for the comparability or the testing of these quality attributes. Therefore, we require them to give us as much as data about the analytical testing of these quality attributes to just prove that the product is biosimilar. Well, that's not just the case that they have to just test their product, but they have to present the data in a systematic way. They have to, there are multiple statistical tools involved. They have to assign some quality ranges of both based on the characterization of the reference product. And they just have to prove that, yes, these quality ranges are accurate and our products fall within these quality ranges. Just have a look. There are like the quality attributes are maybe there are. I told you them. Sometimes there are hundreds of quality attributes which they have to test for their biosimilar products. They have to test the primary structure, higher order structure, glycosylation product related variants, process related impurities, product related impurities, and they have to use a lot of sophisticated and expensive equipments, just like the mass spectrometry, HPLC, electrophoresis, and many other tools which has to be used for the during the development and analytical similarity of these biosimilars. The next thing is, as I told you, that once the analytical similarity assessment is done and you have the satisfactory results, there are always some minor differences we find it during this assessment, especially in the form in glycosylation profile. The glycoforms have the tendency to variate. So if there are some differences appear, then the company address them through the in vitro and in vivo, the non-clinical or preclinical testings. And if they are satisfied, even we sometimes waive this requirement of non-clinical assessment. Then the next part is, the other important part is that up till now, regulatory authority demands two types of clinical studies. First one is the phase 1 clinical study, and the second is the phase 3 clinical study. There is no requirement for the phase 2 clinical trials. In the phase 1 clinical study, the biosimilar has to prove that it is comparable in terms of pharmacokinetic, pharmacodynamic, if the endpoints are clear, and in terms of safety and immunogenicity with the reference product. But in the phase 3 trials, they have to prove that their product, they have to conduct the clinical trials on a large number of population, comparatively large number of population on even the large number of patient population, where they have to prove that their product is comparable in terms of efficacy, safety, immunogenicity with the reference product. If these clinical trials and the clinical studies are successful and they are able to prove that their products are comparable, then that means that they have shown that their product is highly similar through the quality biosimilarity and the clinical comparability, and the regulatory authority on the basis of the total results of all these studies approve the biosimilars. Since these biological products have unique characteristics, these biosimilars are unique in nature. They have a unique structure, they have unique manufacturing processes, they have a newly and unique regulatory framework. Therefore, some challenges are also unique. So I'll just quickly go through some of the challenges which we face, the companies face during the development. The reference product. Well, as I told you, the definition of biosimilar in various regulatory authorities are same. But there is a minute difference in the EMA and US FDA definition, and that is the definition of reference product. The EMA says that the reference product is the one which is approved in EMA, and the US FDA says that the reference product is the one which is approved in US FDA. So therefore, if the company intends to develop a global product, so they have to have to buy the reference products from the both markets, and they have to bridge these studies and conducting on the both products originated from EMA and US FDA and bridge them together. Okay, and another thing is the limited knowledge about the reference products. Of course, these innovator companies are very smart. They know how to hide their information. So you will not find much insight about the basics of these biological products. Only thing you have is the publications, public assessment report by the regulatory authorities, product information, and the only thing that is the developer of a biosimilar relies is the characterization. They purchase a lot of samples from the market, they characterize it, they reverse engineer it in their lab. They know, they try to find that what type of the primary structure, secondary structure, and tertiary structure bear by this reference product. They try to know much about the glycosylation profiles, what type of product and process related impurities are in this reference product, and other critical quality attributes. So they have to spend a lot of money and effort on the getting the knowledge and insight about the product. Cost and budget constraints. Of course, I told you that these biological innovator products or the originator brand can be very expensive. So and for to establish biosimilarity, they have to purchase a lot of different batches and a lot of different samples from the international market, and it becomes very expensive for the biosimilar developers. Yeah, we also expect that because I told you that there is a tendency that biological product, the quality attributes of a biological product may vary from batch to batch. Therefore, we require then that the reference product you purchase should have a varying shelf life and should have from the multiple batches. The regulatory USFD actually defines the exact number of minimum number of batches which are required for the biosimilarity, but other regulatory authorities they said that at least you have enough batches of reference product to confirm that the study you have conducted has a enough statistical power. Formulation differences and excipients. Well, this actually happened with Humira in 2011 or 12, I guess. They changed their formulation from the buffer in their formulation. They changed it from the citric acid to citrate free, and nowadays you find Humira formulation which are citrate free. But at that time, when the biosimilar developer were developing their product, they purchased a sample with a citric acid, and maybe they were at the later stage of the clinical trial or somewhere. Suddenly, when they found that the innovator has changed the formulation, then they have to conduct extra studies on the new formulation to just confirm that their new biosimilars is actually comparable with new formulation as well. So there sometimes such kind of things happen in the regulatory world. Well, patents, of course, this is the biggest fight going on between the innovator biologics and the biosimilars. And they don't only patent their molecule itself, but they also patent the formulation, manufacturing process, and this very specific use too. So it's always a legal battle going on in the court of international courts between these biosimilars and innovator. Just to give you an example, that the adalimumab Humira has been protected by over 100 patents in USA. Therefore, the biosimilars of Humira was first approved in 2007 in Europe, but US FDA took like 6 years more to approve these biosimilars. But they were already approved in US FDA, but launching was made in 2023 due to these patent issues. Manufacturing complexities. As I already mentioned that the manufacturing process is complex, and slight variation in any of the things like the temperature, agitation, pH, or even in the cell line handling may change the quality of the product. Quality of the product means any glycosylation profiles, product-related impurities or process-related impurity, which may impact on the safety and the efficacy of the product later. Analytical similarity assessment, we discussed this. Of course, there are challenges. Obtaining reference product is a challenge. Using the instruments, gaining access to the sophisticated analytical instruments is a challenge. Because there are no standardized method, they have to develop and validate it. High sensitive method has been demanded by the regulators because so that they can show some minor differences in the glycoforms and the post translational modifications due to the inherent variability. And also they have to submit their data by using a lot of analytical and statistical tools. Yes, the regulatory environment is evolving. For biosimilars, it's already been, I think, maybe 24 years has passed since the first guidelines of biosimilars, and we have already approved multiple biosimilars in the international market. So now we have gained a lot of information and insights about the biosimilar. Now we know the exact manufacturing process of a recombinant protein. Now we have more access to more sophisticated and new equipments and instruments which can give us the better insight of their structure and the physicochemical properties and functional basis. So after this, now the debate has been started since then that we have to now reduce the requirement for the regulatory approval for these biosimilars. So currently, the debate is going on that we have to abolish the requirement for the phase 3 clinical trials because they are expensive, and some experts have the opinion that they are like of no use. They doesn't give you the much more insight about the biosimilar. The clinical phase 1 and your analytical similarity assessment is enough and sufficient for establishing the biosimilarity of the molecule. For that, WHO has already issued a revised guideline in 2022, in which they have mentioned that phase 3 is only required when you don't have enough evidence through the previous analytical similarity and the phase 1 trials. UK has also adopted this same WHO concept. EMA has issued a working paper and they are discussing on it that how we can move forward. And USFDA is also in the discussion phase and consultation phase whether we have to abolish the requirement of clinical phase 3 trials or not. Some of the other challenges like interchangeability. Interchangeability is a very unique term only used in the US FDA. They think that if a biosimilar has to prove itself as interchangeable, it has to conduct further studies to prove that they are interchangeable. Like interchangeable mean without the prescription of a doctor, a pharmacist or the healthcare provider can switch from the biological to the biosimilar product. But this concept is not widely applicable. The other countries like in GCC and in Europe, the concept of interchangeability doesn't exist. So it can be the therapy can be switched on the discretion of the healthcare provider. Naming conventions, very unique. USFD has a very unique way of naming these biosimilar products. They usually use the name with the name of the biosimilar product. They use a very unique alphabets, which has no meaning. Like they can be A, B, C, D, W, X, Y, Z, or anything. So they have a unique just to differentiate that these are biosimilars. Extrapolation of indications. As I mentioned that in the phase 3 comparability trials of biosimilar, they don't have to conduct clinical studies on all the indication the reference product has been approved. They just have to pick the most sensitive indication and compare their product with the reference product. And later on, if they have to justify that if the structural attributes are same, biological function of the product is same, non-clinical toxicology is same, human PK is same, and phase 3 is same, then regulators approve the same biosimilar for the other indications too, without further studies. The concept is the biological drift. Nowadays, because as I told you, due to the heterogeneity and inherent variability of these products, sometimes the manufacturer undergoes very minute changes in their manufacturing processes and without knowing it involuntarily, and which is actually affecting the post-translational modificational profile like glycoforms of the biological products, which are ultimately creating the immunogenicity profiling in the patient population, affecting the safety of the product and the efficacy too. So this is also the challenge which in the current world, in the commercial manufacturing, the regulatory authorities are about to look for. I think I have finished this presentation in 45 minutes. That's a success. And thank you very much, everyone. I tried to include as much as information, which actually at this stage, what I think is required. And if you have any questions related to anything about the biosimilars, you may ask.
Thank you so much, Dr. Adeel. If anyone have any question, they can raise your hands or type in the chat box. Dr. Adeel, we have one question. It's from Amber. I think so, yes. It's from Amber. Larger group of population means for phase 3 comparable data. Minimum sample size should be.
Look, for these kind of a product, there is no minimum sample size. In phase 3 trial, we assume that it should be like maybe in thousands of, hundreds and thousands of patients. But since these biological product and biosimilar, mostly you see they are for the oncology therapeutic group or immunology, where you have a very limited number of patients available. Therefore, for these, because I told you it's a stepwise approach. You have to consult for everything during the development time with the regulatory authorities, and with then you have to devise the study design. So if for these kind of biosimilars, you have to, the study design is agreed with the regulatory authority only when it has enough statistical power. Maybe and depending on the availability of the patient population in the region. So there, it is totally customizable, and it decided with the consultation with the regulatory authorities.
Thank you, Dr. Adeel. Any other questions from the participants? Please raise your hand. We have just 5 more minutes. I think so. They have no question. I have one question, Dr. Adeel. What are the
Yes.
legal risks associated with biosimilar development and the commercialization?
Legal.
Yes, yes, the legal risk.
The legal risk is mostly, I told you that the exclusivity and the patent issues. Uh, the Most of the time we see a war going on between these biosimilars and innovator products. So the only thing that ties the legal constraints are because the innovator has so many patents. I told you that it's not only related to the specific molecule, very small things related to the formulations, excipients even, the process they have, or the cell line sometimes. So these kind of issues may arise later on. But you see that most of the biosimilars approved in the international market are actually goes through the development process either in Europe or the US FDA. So these legal things are being catered earlier before giving them the marketing authorization.
Thank you so much, Dr. Adeel. If we have any question, I think so. We have no question. Thank you so much, Dr. Adeel, for your valuable time and very impressive presentations by you. And I have a request from students. Please fill the feedback form so we can share the upcoming webinars to you. And once again, thank you, Dr. Adeel. Thank you so much.
Thank you
Thank
very
you
much.
everyone for joining.
Pleasure
Thank you.
meeting you. Thank you very much.
Thank you. Thank you so much.